Bharat, you're using phone. Not allowed in class. See from camera and take your phone. Give your phone now. Give. Play games in class. Oh no, give. So secretly using phone. Why not come you? Washroom. Now time over. Sit. Now no editing in class. No headache, head, do nothing. Sit. Study. Sit. No. Okay, go. Exactly. हाँ यहाँ पर सबसे 520 की लेक्चर अभी तो शिंदे था हाँ नहीं आज तो शिंदे हाँ विंडोज लेकिन 520 के लेक्चर होता है चलो अभी सब कुछ स्टार्ट हो गया क्यों अंश तुझे पता है तू एक जस्ट टाइपिंग पर बैठा है भाई मैं हटा दिया उसको नो खुद भी डेढ़ सही हो ठीक है मैं इधर मेरे से साइड में इधर अभी देख लोग तेरा सबसे मारो अच्छी हां नहीं बस जानना भी है अच्छी बात नहीं बता रहा इस भाई तो है कल लगे बैग स्टोरेज का एकदम कैसा है कैसा है यार कैसा है कैसा है फुल सारे लोग सब साथ में रहे हां आज का तो थोड़ा सा तू सही आज का ही नहीं अभी तो बच्चा बैठ रहा था कब से घूम रही थी अभी फिर ये सही कर रही है तू गई थी क्या घूम ही नहीं आ गया भाई फिर जाने वाली सामने आके तो कैसी होना है वाह इतना You are into it. Now they need to build a digital touch base. Yes. Do the fact. No problem. नहीं तू नहीं okay है तू नहीं है तू तो करने वाला है तू जब भी हो जाए तो भाई सर ये बैठता है बहुत गंदे का दिखता है देखा देखिए तू फिर घूम रहा है तू पंडाल में उसके अंदर किया Wait, I took that person. हम दोनों नीचे तो टेंशन बैठना ही देख हमारे पास ज़्यादा नहीं मैं तो हमारी चाहिए अभी तू मेरे तू उसके तो पता नहीं क्यों नहीं I know but मैंने उसको invite भी किया था सामने से चल क्या नहीं बाजार जो मेरी है तो अबे यार वो कल लोग मेरा क्वेश्चन पेपर और आंसर पेपर ए हमारे के घर पे तो कल हम को बोला था 2:30 बजे तक हमें बोला खींचा था टाप उठ लो मैं जाएगा तुम्हारे प्रोग्राम में हाई चल 4 लोग ऑलरेडी खा के आ गए 4 फिर ठीक आउट कर अंश चाट का खा के नहीं खा ठीक और चाटा है और मेरा क्वेश्चन पेपर मतलब कि बाहर वो भी वेट कर रही है हां तुम लोग प्योर वेज से खाना तुम हमेशा
अभिप्राय नहीं आया यार आज अंतिम नहीं वो
नहीं एक्साइट because the rules are also active or if you want to active
no like no we
if they are not fertilized then they are veg and most of them are yes sir
बन गया और ऐसा स्टेट लाइन
आधी रोज एक डिश बन जाता है मजा नहीं उसको पता नहीं है उसको इस बात का नहीं उसको इस बात अच्छा वो मुझे तेरा आंसर बेसिकली मैंने आपसे पूछा मालूम किया कि नेचर की
मेरे मन में सही सा गाना
appreciating qualities of nature
दो टाइप का क्वेश्चन कोई सुनाता दिया आपसे मतलब अपनी नाम की नहीं सुना होगा
बोला था 5 मिनट के काम 10 मिनट देवांशी मैंने बोला था 5 मिनट अरे अच्छा देंगी मैं तो सुना नहीं
किधर बैठा
पेंसिल चल
अभी नहीं मैंने इसका डीएम नहीं करूंगा पक्का क्या करा था नोटिस में
इसको क्यों
तुम्हारे साथ ही है क्या भूल से भी ना
मुझसे क्या लेते
जा मैं तो हर
शक्ल इनके लगता है
Now I will take you a compliment or
लिटरली तुम लोग वही सेम गेम में रहते 60 प्लस 40
जिन हम जिन के
भाई तो जागरण
अरे वो हम लोग ने डिसाइड किया था फिर क्या होता है सेक्रेटरी बताओगे सेक्रेटरी आती है पोती नो दिस इज द पब्लिक हॉल यू कैन यूज इट दिस 20 नहीं this is a banquet hall just 20 foot and AC cannot be working for just 5 of you this is the reason अरे वो सेंट्रल नहीं दोस्त तो फिर उससे आगे क्या होता है हम लोगों को बोला नहीं क्या कहा जाए फिर मैं बोलते पड़ी चलो खाली पड़ा मैं हम पढ़ने जाते हैं उसके बाद फिर मैं फिर वजन निकलता जागरण का कुछ जागरण का नाम ले कुछ जागरण नाम ले जिंदगी का करना तो जब बिना मैं तो आगे बता आई तो
Sit. Okay, now take out your notebooks. We'll just do 2 questions and after that we'll move ahead. Take out your notebooks. Take out your notebooks. Yes. The first question, right? You will try answering now on your own. Take out your notebooks and write down the first question. The first question.
I missed one class.
Why? But
मैं sorry ma'am I'm late ma'am may I come in ma'am sorry ma'am
तुमने जैसे बहुत बोला
मैं तो already था
और तुम तो हमेशा लेट आते हो. Today is the first day when you are already inside the classroom. Otherwise, your entry is always gallantly will enter the classroom. You did not check the message. You and your name is Aditya. Aryan. How come you both did not check the message?
I was unable to put my message outside from 12 class.
Chalo. Number 1 is, write down. What is the significance of and notebook है? तो रफ बुक तो लाए हो ना something you have no with you, such a big bag you brought. Notebook नहीं है, रफ बुक भी नहीं है, कुछ नहीं है. Go and get अच्छा paper लेके आओ जाओ. Go and get some extra sheets of paper. Go fast. Fast. Number 1. What is the significance of? Write down the name of the chapter. You'll have to solve some questions. Many questions you'll have to write also. Number 1. What is the significance of? What is the significance of the concavities of RBCs? Significance of the concavities of RBCs.
Increase in surface area.
हाँ, देखो complete answer लिखना है.
And easily flexible and easily squeezed through.
That is because of the small size. That is not because of concavities. देखो. What is the significance of the concavities of RBC? Number 2. Concavities. You may leave 3 lines for your answer. Go to the next. Number 2. Go to the next. Number 2. Under what conditions does the RBC count increase in an individual? Under what conditions does the RBC count increase in an individual? Increase in an individual.
How many marks?
These are all 1 mark. Your entire paper is 1 mark question. Next, number 3. Number 3. Name the temporary compounds. Name the temporary compounds. Temporary compounds formed. Name the temporary compounds formed by the combination of hemoglobin with. By the combination of hemoglobin with. Put a dash. 1 oxygen. Next line 2 carbon dioxide. Next line 3 carbon monoxide. 1, 2, 3. I'm asking you to write.
Why are they temporary?
Because as the situation reverts, the compounds will break again. At different parts of the body, the compounds will break again. They are not permanently formed.
Carbon monoxide is stable or something like that.
No, not stable. It's that only that the hemoglobin has got a more affinity for carbon monoxide. But even if a person who is drowning, if you can relieve that person very soon, then again that compound also will break. No talking.
Found with hemoglobin.
I want you to write all the 3 compounds. Number 1, combination of hemoglobin with oxygen, carbon dioxide, carbon monoxide. Carbon monoxide. Number 3, what is hemin? Number
2 more questions.
all 1 mark. तुम्हारे पेपर में 2 mark question है कहाँ? The entire thing is 1 mark. Hemin. Section A, if you check the entire section A is 1 mark. Section B also majorly it's 1 mark question. Okay, there. Ansh, idhar aao.
Please.
Number 2. I said number 2. What is human? What is human? H-A-E. Check the spelling. Last, leave a gap of 2 lines. Number 4.
Human is protein.
Human is not protein. Human is not protein. Next, 2 differences between blood and lymph. Number 4. That's 5, right? 5. 2 differences between blood and lymph. 2 differences, blood and lymph. Chalo, fast. Then we'll move ahead with the next part of the chapter. Fast. Try writing on your own. Aditya, both you stop talking and try making an effort to write on your own.
I wanted the first and second question.
First question क्या है?
Concavities of RBC.
Significance of the concavity of RBC. Just now we discussed, you should be able to write all.
Surface area ratio was there. More oxygen will come
More oxygen will be absorbed and delivered to the tissues.
Second one mark.
Only 1 point only. Only 1 point. Just 5 minutes I'm giving you, so don't waste your time. Finish your work.
Ma'am, another point I was something like more RBC.
That is a small size. What is it?
Making heard noises.
Ansh.
Please.
Anshi will show me all the answers written. I want to see what you write. Under what conditions the number of RBC increase?
High altitude.
2 conditions likho.
And
Minimum
polycythemia.
2 conditions you write.
High altitude, 30% increase. Polycythemia and
High altitude, because of high altitude it will increase, the number of RBC will increase because of the high altitude. What else I discussed just now?
Polycythemia.
Under what condition the number of RBC abnormal increase, that is a persistent increase, no? Temporarily, under what condition will the RBC increase?
Drowning. That is carbon monoxide.
Under what condition does, for a short period of time, in certain stages of life, in pregnant women, during the phase of pregnancy in women, for a short period, the number of RBC is increasing. In infants, during the process of growth, short period it is increasing. Not for a permanent abnormal. Now you cannot write about polycythemia. This is a persistent condition. Write down the name of the compounds correctly.
Even is the iron
Containing part of hemoglobin. Iron, that's a question quite often you get in your paper. Iron containing part of hemoglobin. Then the cell organelle, deficient cell organelle, and the significance, that also you should read properly. I did not give you questions as such now, but that part also you should read properly. Done?
Done.
Got the answers, all of you? All of you got the answers? Aditya got the answer, the first one. What do you wrote?
More absorption of oxygen and more surface area and greater volume. What is the significance of concavities of RBCs? More absorption of oxygen and more surface area.
The concavities help in what? More
absorption.
surface area and hence absorption of oxygen. Frame your answers properly. If you don't frame your answer properly, I have to give you quite a lot of writing, because if you don't frame your answer properly, you will not get perfect mark. Next, Ansh, what you wrote? Next answer.
I didn't get this. I didn't get this.
What is that question?
Name the temporary compounds.
3 temporary compounds with hemoglobin. 1 with oxygen, what is it?
Hemoglobin.
Right. Second, Aditya, what is it? No, the temporary compound, that question. With oxygen, it is oxyhemoglobin next.
Oxygen, oxyhemoglobin, the carbon dioxide one
Carb amino hemoglobin, and carbon monoxide carboxyhemoglobin. Write down all these three words should be very clear. Next, the third question, what is it?
Difference between blood and
What is the third question?
Him was the fourth question. This was the third question.
This was the third. Okay, human we already discussed, iron containing component. Next is
Blood and lymph differences.
blood and lymph difference. What are the differences to write? Bolo. What are the two differences to write? Blood and lymph. Blood and lymph in the paper you will get composition, function also. But the later part we are going to study lymphatic system also a little bit. So, filhal, whatever I had discussed. Let us talk about the composition only. Two differences.
Okay, so the blood contains RBCs, WBCs, platelets, and plasma, and the lymph contains platelets and plasma only. And WBCs.
No, platelets not there. Lymph faint. Platelets is not a major component to ever write. Lymph has leukocytes and plasma, WBCs and plasma. Okay. Color, red color.
Tonsils are found here.
Red color for blood.
Tonsils. So if we hurt them, white color fluid will come out.
No, why?
Because lymph is there.
No. No.
Why
When,
colorless?
no. Second point. Second point. What is the second point? Blood is red in color, that is colorless. Blood flows in blood vessels. All of you please write down some few points. See that you write your answers very nice. Blood flows in blood vessels, lymph flows in lymphatic vessels. Function also, we need not wait for the last part of the chapter today to reach there. You already studied this earlier. Functions of blood, if at all to write, what are one or two functions? Just now we have been talking about transport of oxygen, carbon dioxide, transport of nutrients. For lymph, is there anything extra that we had studied earlier?
It helps in fighting off infection.
वो तो blood के साथ common है. Is there any other separate function of lymph you had studied earlier? Lymph helps in defense, of course, because it has got leukocytes and absorption of fats.
Lacteals.
You had studied last year, digestive system. So absorption of fats. So please write down two structural and one functional difference. So composition you write, blood vessels, lymphatic vessels you write, blood vessels, lymphatic vessels you write, and function. Blood helps in transport of oxygen, carbon dioxide, distribution of nutrients.
Ma'am, but in the starting you told us that the limbs, the vessels come into the vein or something like that. So the lymph is also in the blood vessels, right? Kind of.
Lymphatic vessels are separate, but the lymphatic vessels, there is no separate lymphatic system in our body. No separate vascular system as that. But when we are talking about the blood vascular system, in the blood vascular system, there is a series of tubes, which is the lymphatic system. But they are not independent system in our body. So they will always go and meet the vein.
Yeah, so
meet the vascular system again.
we cannot write the point of difference as like the presence in blood vessels and heart, because in the blood vessels also lymph is present. Technically, it is present in.
No. Blood vessels, to mention about vessels, if at all you get a difference, blood and lymph within bracket vessels, the parameter given, then you have to write blood flows in blood vessels, arteries, veins, capillaries. Lymph flows in lymphatic vessels, lymphatic channels. No arteries, veins to mention there. It just goes and meets as a phase. The next type of cells, we are talking about the blood. The next type of cells, all done, no? Questions all done?
Yes.
Now the next type of blood cells that we are talking about is the WBCs, also known as the leukocytes. Leukocytes, from the term itself, white, colorless leukocytes. Then, same like that, when we are talking about the major features, what are the major features of the leukocytes to understand? If I have to talk about the origin again, the origin, bone marrow, same like the RBCs we have talked about. But something more that we try understanding, since it is the WBCs we are talking about, WBCs are formed from the spleen also and from the lymph nodes also. We have talked about the lymph nodes in the previous speaker. I was talking about this. Lymph nodes, what are these? These are small nodular structures all throughout the lymphatic vessels where the lymph can accumulate for a short period of time. In the flow of the fluid, lymph remains accumulated, and that particular nutritive region can also give rise to blood cells, the WBCs that we are talking about. Now, WBCs again, some other features to understand. WBCs, the structure, if you have to understand, what about the basic structure of the WBCs? The basic structure of the WBCs, do they have a well-formed nucleus? Yes, they have got well-formed nucleus. You can see the WBCs, they have got well-formed nucleus. There are 5 different types. They have got well-formed nucleus. Nucleus present. Nucleus present, and based on certain features, characteristics of the nucleus, they can again be categorized into 2 main types. What are the 2 main types? One is the agranulocytes, and the other is the granulocytes. The agranulocytes and the granulocytes, the 2 major types that we are talking about. Agranulocytes, from the concept that the cytoplasm, which are the agranulocytes, if you see, there are certain type of WBCs which does not have any granular substance in the cytoplasm. So agranulocytes, and a number of them, or 3 major types, they have got granular substances in their cytoplasm, which can take up a particular stain, which can take up particular chemical substances. So these are known as the granulocytes, agranulocytes and granulocytes, the 2 major types. So now when we are talking about the agranulocytes and the granulocytes, and which are the type of WBCs and what are their specific characteristics to understand. If I talk about the granulocytes, also known as the granular leukocytes. Which are the granulocytes? The granulocytes are the neutrophils, the eosinophils, and the basophils. 3 types. The neutrophils, the eosinophils, and the basophils. Few features to remember for each one of them. The neutrophils. The second is the eosinophils and the basophils. The 3 types. So now, when we are talking about the neutrophils, yes, features are what? Neutrophils, they are granular leukocytes. What happened?
No, you said basophils. In sense organs, the bacillary membrane, so I confused you with it. I'm just.
Sense organ mein tum gaye kyun?
I was reading it yesterday for the exam.
So now, when we are talking about the 3 types, granulocytes, what are the 3 types? Number 1, neutrophils, eosinophils, and basophils. So neutrophils, they are the maximum number of WBCs. The type of WBCs which are maximally present in our blood is the neutrophils. Almost about 60, 65, 70% of the WBCs are just neutrophils. They are granular. Now, specific features when we are talking about, see their lobes. Quite a number of times you are given this type of images in your question paper, and you'll be asked to identify on the characteristics questions on them. How many lobed? The nucleus, when we are talking about, do they possess the nucleus? Yes, they possess nucleus, but a number of their nucleus will be lobed. They have got masses connected by the chromatin threads, if you see. That's the reason the nucleus is known to be lobed nucleus. The nucleus is lobed, a major feature. So lobed nucleus, always a feature for WBCs. Now based on that, neutrophils, if it is 2 to 3 lobes, 3 to 5 lobes, I'm sorry. If it is neutrophils, it is generally they will be having 3 to 5 lobes, always. Talk about the next one, eosinophils. Which one is the eosinophil? See here, eosinophils. Can we understand the lobes? Yes, the lobes are distinct, generally 2 to 3 lobes. If it is eosinophils, how will you identify the figure? 2 to 3 lobes. If it is basophils, lobes are there, but it is indistinct. The lobes are not clear. The lobes are indistinct. So that is one feature how you can identify the cells. So this is 2 to 3 lobes, and here it remains indistinct. It is not very clear. That is one feature of identifying them. What are the other features about each of these cells? Now, generally, whenever we talk about the WBCs, what is their role? Their role is always defense. But what are the two methods by which the WBCs provide defense? There are certain methods that you'll have to remember. So I talk about the neutrophils. What is that one specific defensive method that always should be remembered with neutrophils? That is phagocytosis. Neutrophils and phagocytosis. What is the meaning of phagocytosis? This type of a cell has the ability to engulf the disease-causing germs, just like an amoeba. Just like the process of method of nutrition in an amoeba, how does the amoeba take in the food? Pseudopodia. But here the concept of pseudopodia is not that very prominent. But then these type of cells, whenever the germs and the pathogens are near them, they can generally form a cup-shaped structure, take in the germs and destroy. So that's a concept known as phagocytosis. Come to the next one. Now, when we are talking about the eosinophils, now this eosinophils, this come in the category of another type of a Anshu, come here. You are writing nothing in the paper when I gave you the answer. Did you write?
Yes, ma'am.
I had asked you one answer to tell me that you could not tell me.
I wrote.
Come here.
I mean, put that in my bag.
You come here. Come here straight. You get them in the school, no? They're in your same school?
Mommy kept the trash in my bag.
No nonsense in my class. Come this side and sit. Now, when we are talking about the eosinophils, what is that property? Now, here, all the 5 types of WBCs, you will have to remember what type of defensive mechanism associated with them. So, I said, if I talk about based on the lobes, yes, that is very, very important because for identification of the diagram, it's very necessary. Which type of cell, how many lobes, what about the nuclear structure? So, first of all, we are talking about the granulocytes. Eosinophils are associated with one type of a defense mechanism, that is, they start releasing special chemical substances, and these special chemical substances are known as antitoxins. Antitoxins are special chemical substances always released by the eosinophils. Okay, what about the basophils? So when we are talking about the basophils again, they are also associated with defense, not by eating the germs, but they also start releasing some special chemical substances. And what are those? They would start releasing special chemical substances known as the histamines. So now when we are talking about this major 3 types that I'm talking about, neutrophils, eosinophils. Eosinophil, the number of the eosinophil always will be high whenever there's an infection in our body. Eosinophil blood count, in the blood count, the number of eosinophil high always indicates infection. What about the basophils? Whenever again there's an infection leading to some inflammation, for example, we are bitten by some insects, mosquito bite, and then there's an infection, there's an inflammation, no infection as such now, we are talking about an inflammation. At that time, this cell increased in number, releasing the histamine and neutralizing that inflamed region, inflammation. So here we are talking about the defensive mechanisms, 3 types of cells. What about the other side? The other side, when we are talking about granulocytes, and the other is agranulocytes. Agranulocytes. Now, which are the agranulocytes? What is the major difference? Granulocytes and agranulocytes. Within bracket, you get cytoplasm. Based on the cytoplasm, you have to write. Very simple. Cytoplasmic granules absent, cytoplasmic granules present. If the cytoplasmic granules are present, they are very easy to be stained. They take up specific type of dyes. Dyes meaning stains that can be used to see them under the microscope. The neutral dyes, the acidic dyes, the basic dyes, like that. Agranulocytes, which are the cells? 2 types again, yeah? One is we are talking about the lymphocytes, a major type of a WBC. These are very, very important types of WBCs. And then we are talking about one more, that is the monocytes. Now, what are the special properties for each of them? I talk about the lymphocytes. How will you identify? Lymphocytes can be smaller, larger lymphocytes. To identify the lymphocytes, when we are talking about the basic structure, their nucleus would be sometimes spherical also, sometimes bean-shaped also. When we are talking about the monocytes, the nucleus is quite large, spherical mostly. See the diagram in your book also. The lymphocytes, the monocytes, characteristic to identify. The lymphocytes will always have small lymphocytes or large lymphocytes. The nucleus is quite big. The nucleus will be quite big. Now, what is the defensive characteristic? Defense how? How do they carry out the defense? If it is the lymphocyte, they will be releasing special chemical substances known as antibodies. These antibodies are always released when an organism, when an individual suffers from an infection or vaccination. So these are those type of cells that I am talking about, the lymphocytes, which will be releasing the antibodies.
Ma'am, antibodies is the next page. I was just reading this. Antibodies act as antitoxins, it is written. So is there a difference between
antibodies and antitoxins? There is a slight difference. If I talk about, for example, a bee sting and poisonous chemical entering the body, then it will be the antitoxins which can neutralize. It will be the antitoxins which can neutralize. But at the same time, if it is a germ which has entered the body and bringing about some changes, not allowing the cell function properly, then the lymphocyte will release the antibodies and antibodies can neutralize the entire infection. Monocytes, same here. How do they bring about defense? Monocyte also show the process of phagocytosis. So name the two types of WBCs which show phagocytosis. Neutrophils, monocyte. This is a question again. Difference between these types of cells based on their defensive mechanism also is asked. So neutrophil, always phagocytosis. Antitoxin with which type of a cell? Then histamine with which type of a cell? And then antibodies with which type of a cell? You will have to remember this. Now, since we are talking about this property of the defense mechanism, let us try to understand what are, in general then, what are the two methods by which WBCs provide defense? Either by phagocytosis or by releasing special chemical substances. What are the special chemical substances that the WBCs can release? Antibodies, the most important, histamine, antitoxins. So these are the three chemical substances that they release, or the major two types of cells, neutrophils and monocytes, also can eat away the germs, engulf them and destroy, a process known as phagocytosis. Now, something more that we are talking about, that next when we are talking apart from the cells, next is we also come understanding that what would be some other features. Now, this WBCs, we are saying that they are capable of phagocytosis, but how will they be capable of phagocytosis? I may talk about a blood vessel and then the blood here, the WBCs are comparatively bigger and they are supposed to have a lobed nucleus. So if this is the WBC there and if this is the capillary pore, I understand. Surrounding the capillary pore, what it is? Surrounding the capillary pore, the body tissues are there. So talking about a specific feature that WBCs show phagocytosis. How can they show the process of phagocytosis? Because this WBC can change its shape and it can become extensively long and can squeeze in through that small capillary pore. This particular property that the WBC show, what is it known as? This is known as diaphadesis. What is this? This is known as diaphadesis. What is the concept of diaphadesis? A process by which the WBC can squeeze in through the small capillary pore. But how can they do that? Because they can change shapes. So what is that one special property of the WBC? The WBCs are amoeboid. The WBCs are amoeboid. They can change shapes. Similarly, we talk about that if I would have drawn one RBC here, the capillary pore remains the same, but I draw one RBC which is much smaller than the WBC. What about the RBC? In all those concepts so far I have been talking about, the oozing out of the plasma and all those concepts to form the lymph, never we had come across that the RBC oozes out or comes out through the capillary pore. Why the reason? Because RBCs, they are not able to change their shapes. They are not amoeboid. The WBC is a amoeboid and hence the concept of diabetes. So what is the definition you write down? What is diabetes? WBCs can change their shapes, squeezing through the capillary pore and reach the site of infection. Wherever the infection in the tissues, they can reach the site of infection and carry out the process of phagocytosis. So this relation has to be very clear. Amoeboid, phagocytosis, diabetes, how are they all related with reference to the WBCs? Then next is the number. If you have to talk about the number of the WBCs, origin, how are they formed, we had discussed. The number of the WBCs, what was the number of the RBC we discussed? In millions it was, no? Now for the number of the WBCs, it is 4,000 to 8,000 or 9,000 generally. But we are talking about cells, not million. We are talking about cells, 4,000 to 9,000 cells per cubic millimeter of blood. Wherein in case of RBC, we were writing in millions, 4 to 4.5, 5 to 5.5 million cells per cubic millimeter. And here the number is much lesser. Okay, 4 to 9,000 cells only we are talking about. Is there any chance where the number of the WBC increases? Yes, the number of the WBC increases whenever there's an infection. When the body's defense mechanism need to be active, at that time the WBC increases. The number of the WBC again may decrease whenever somebody has suffered from a surgery or childbirth, etc., etc. These are the reasons where the temporarily, for a short period of time, the number of the WBC may decrease. Then, if there is a persistent increase, same like RBCs again, we talk about, if there is a persistent increase, persistent decrease, is it possible? Yes, there's a condition where the leukocytes, if there is a persistent increase, abnormal increase that we are now talking about, if there is an abnormal increase, what is the abnormal increase known as? The abnormal increase will be known as the leukocytes. What are the WBCs known as? Leukocytes. So we are talking about a condition, abnormal increase will be known as
abnormal increase is not given.
Huh?
That is, leukemia is not abnormal increase. That is some cancer.
Abnormal increase in WBC only, that is. When we are talking about leukemia and the abnormal decrease, these are the words you have to remember. Abnormal decrease. What is the doubt? Tell me.
Ma'am, in the next page, like the just phagocytosis paragraph, there it is written an abnormal decrease up to 50,000 something. I just saw the number. So, like what my doubt is, leukemia, what I understand is like a type of cancer or something.
Cancer, which leads or which is because of the abnormal increase. That is actually typical blood cancer when we talk about. Blood cancer that people talk about or you know about, that is all about the increase, not in the number of the platelets of the RBC. Always in the increase in the number of the WBCs. It is abnormal increase in the number of the WBCs. So take a note of these two words. Leukemia, abnormal increase. Leukopenia, abnormal decrease. For RBC, what was that word we were talking about? Abnormal increase was
Polycythemia.
Abnormal decrease was
Erythropenia.
So for WBCs, for RBCs. So here, what are the average number? What is their average number? That is very important. The types, the 5 types is very important for you to know. What are the 5 types? How will you identify the 5 types? How will you identify the types of the cells? That is very important. Identification of the types of the cells very important, and the features. The last type of cells that we are talking about is the platelets. Now platelets, nothing much that we have to talk about. Platelets. Now I'll just show you one image to understand that these platelets are actually also not well-formed cells. See here? The platelets are not well-formed cells. Actually, when we are talking about the platelets, they are also known as fragments. So, the three major types of cells, we have been talking about the RBCs, we have been talking about the WBCs. Now, the third type of cells known as platelets. What is the other word for platelets? Thrombocytes. What are the common features that remain the same for WBCs and RBCs? Their site of formation. Again, here also the bone marrow. What is the site of formation? Site of formation remains the same, bone marrow. Now, for this, we were talking about also the lifespan, right? We were talking about the lifespan. So, lifespan, when we are looking into the lifespan of RBCs, what was it? 120 days. Now, lifespan, even for this type of cells, the lifespan of WBCs are few weeks. The lifespan of the thrombocytes are just few days. So, when we are talking about 3 to 5 days, okay, 1 to 2 weeks for the WBCs. So, here also the process, and here for platelets, only basic 1 or 2 concepts you are studying, that is its function. What is the function of the platelet? Now, that is an important part of your chapter there. The site of origin, same as RBC, WBC. Shape, nothing in detail about the shape you are studying. It is a irregular shape. The shape is irregular. Does it have the nucleus? No, it does not have the nucleus. What are the other similarities with the RBCs or the WBCs? With the WBCs, it has got irregular shape. It does not have the nucleus. Does it have any pigment? No, it does not have a pigment. Similarity with the WBCs. So, major function now, if you now talk about, what is the major function of this cell then? What is the average number? 2 to 4 lakhs. For WBCs in thousands, this is in lakhs, 2 to 4 lakhs per cubic millimeter of blood, that is the number. RBCs in millions, that we talk about. Here, for platelets, the most important thing is to understand what is its function. What is its function? What is the role of the platelets? Platelets always help in stopping the oozing of the blood whenever we suffer from a cut or an injury. Whenever we suffer from a cut or an injury and the blood vessels are torn, the blood starts oozing out at that time. How is it possible? Normally our body starts going through certain chemical reactions, and because of those chemical reactions, the oozing of the blood normally stops. We suffer from a cut, we suffer from a wound, an injury happens, the skin is torn, the blood vessels are damaged at that site. But few minutes to, at the most, if it is a big surgery, etc., then additional medical help and chemical substances definitely required. But if I talk about small cut, wound, etc., certain processes which normally takes place because of the platelets and a series of chemical reactions which will happen at the site where the blood vessels are damaged. And this process of stopping the oozing of the blood is known as clotting. The process of blood clotting. Now, what are the sequence? What are the events? Now, this is very important for us. What are the events of the process of blood clotting? We start with the platelets at the site of the injury, wherever the injury has happened, wherever the blood vessels got damaged. Now that the platelets are damaged, the platelets start releasing certain chemical substances, and then the sequence of reaction starts. Now, since this sequence of reaction, one is related to the other, that's the reason this sequence of reactions are known as the cascade reactions, meaning one component activates the next. The product of that activates the next step. So, this is happening in a chain. So, that you'll have to learn in detail. So, let us write down what are the sequence of reactions that takes place whenever we are talking about the process of blood clotting. All of you will have to write these reactions in your notebook. And these reactions, without any mistake, whenever you are asked, you should be able to write it. This is quite a number of times a complete 2-mark question also in your paper. Whenever it is a section of 5-mark questions, 1 mark, 2 mark, 2 mark. So that can be, or that is also a number of times a 2 mark question. So let us start. Talking about the entire process of blood clotting. So the first is the tissue injury. Whenever there's a tissue injury, which type of cell will start its function? We talk about the platelets. Whenever there's a tissue injury, the platelets will release an enzyme. Which enzyme? This enzyme is known as thrombokinase. Also, it is known as the thromboplastin. And also blood clotting factor 10. X or 10, because it is a series of 12 factors. So when we are talking about the injured tissues at the site of injury, the injured tissues, which cells come into play now? Which cells are active now? The platelets. And the platelets will start releasing the thrombokinase. The thrombokinase, what are the alternative names? Thromboplastin or factor X. Now that thromboplastin is there, where is thromboplastin there? In the plasma. Where is thromboplastin there now? In the plasma, released by the platelets. Now that the thromboplastin is there, another component, another protein which is present in the plasma, which is in the enzyme. Thrombokinase or plastin is an enzyme. So now that thrombokinase is there, another protein which is inactive. What is it called? Prothrombin. This is in the inactive state. This prothrombin, in the presence of thrombokinase, starts getting activated. And what is the activated form known as? This is known as the thrombin. So for the formation of the thrombin from prothrombin, calcium ions also required and vitamin K also required. That means vitamin K and calcium ions both has to be in adequate amounts in the blood.
Thrombokinase acts as catalyst.
Thrombokinase acts as an activator. In this cascade reaction, we don't talk about this as a catalyst. It's not an enzyme, but it is an activator, cascade. Prothrombin gets converted to thrombin. So now, thrombin is the active form of the enzyme. Because now thrombin is there, again remember, where is thrombin present? Where are these reactions taking place? This entire reaction is taking place in the plasma. This entire reaction is taking place in the plasma. Thrombin, now that thrombin is there, another protein in the plasma, fibrinogen. We have talked about the 3 prominent proteins in the plasma: albumin, globulin, fibrinogen. Now we talk about
vitamin K.
That is vitamin K. And then we talk about one more very important protein now, that is fibrinogen. This fibrinogen, which is in the soluble form, I already mentioned all these proteins are in the soluble form. Now this fibrinogen in the plasma was in the soluble form, and now that this thrombin adequate is present in the plasma, this is converting the fibrinogen from its soluble to its insoluble form. Converting the fibrinogen to fibrin, that is the insoluble form. This insoluble form now starts making some network-like structure at the site of injury, wherever the blood vessels are broken. Wherever the blood vessels are torn, at that site, this fibrin makes a network. It makes a network. So there is a network or a meshwork also, it is written as network with what? With the dead RBCs also. There's a network been formed with the RBCs. At the site, the RBC is also getting stuck and everything. So there's a network being formed. and this now in the presence of oxygen becomes hard, and that is what is known as the hard clot. So in fact, when we are talking about this, this is also known as the soft clot. And when it starts reacting with oxygen and now it becomes that blackish skin on the surface of that blackish patch on the surface of the skin, that is what is known as the hard clot. So what are the steps we are talking about for both these steps? Here also calcium ions very much required, and vitamin K a must for prothrombin to thrombin. So what are these reactions we are talking about? The entire sequence of clotting of the blood. The first step, thrombokinase released by the platelets. Thrombokinase activates which step of blood clotting? Prothrombin to thrombin. The presence of the thrombin in the plasma activates which step? Fibrinogen to fibrin. Now that I talk about this particular step, now say suppose I talk about this particular step, the soft clot. You have suffered from a cut or a fall, an injury, and then on the surface of the skin at the site of the injury, you observe that there's a brownish colored patch getting formed, the soft clot. And what you do is you happen to peel it off. You are just peeling it off. Often you must have done that. You happen to peel it off little bit and you get to see that there is a yellowish fluid coming out now. Meaning the process of clotting is almost done but not complete. So that yellowish fluid, what is that yellowish fluid? That is actually the plasma. That is the fluid part, the plasma. But that fluid part, the plasma, does not have fibrinogen. Actually, plasma is supposed to have fibrinogen dissolved. But at this part, if I talk about the fluid, the yellowish colored fluid, that is actually the plasma without the fibrinogen, and that is what is known as the serum. What is serum? Serum is the plasma in which fibrinogen has been removed. Why removed? How removed? Because fibrinogen is all used up in the process of blood clotting. Okay. So now that we are talking about what is the difference between the serum, but then is it the same as the pus formation? No, this is not the same as the pus formation. Pus formation also may happen at the surface of the skin, but due to some bacterial infection. So when this serum is tested and when this pus is tested, this serum has got plasma, no fibrinogen in it, no blood cells in it. And if cells are there, then also the dead WBCs and the RBCs. What about this? This has got enormous microbes in it. This has got enormous microbes in it. So the yellowish colored fluid that is developed in a premature clot is not pus. Justify. What will you write? It is not pus because it does not have any microbes and it has got only the plasma from where the fibrinogen is removed out. Clear there? So this particular concept, what is serum, you'll have to remember here. The process of blood clotting, the steps. Since we are talking about platelets, one concept we'll have to remember, and this is a blood-related disorder. Did you ever come across this? Hemophilia. You had come across this, no? We have finished genetics?
Yes.
Here?
Yes.
Here we have finished?
Yeah.
Now, hemophilia, that is a sex-related disorder. X-linked inheritance we had studied. What was the cause for this?
Recessive X-linked disease.
A presence of
Recessive genes.
absence of a recessive allele. Absence of a recessive allele. And we have come across what is the major symptom of a person suffering from hemophilia? The person profusely would bleed from a smallest cut also. From a smallest cut, the person would bleed profusely. Now this person is profusely bleeding. Why? Because the factor 10 is absent there. The factor 10 is absent in his or her blood. The person is not having factor 10 in his blood. Why no factor 10 in the blood? Because that recessive allele is absent there. So because that recessive allele is not there, the counteracting allele is not there, That's the reason one recessive allele is producing a deficiency in the body, and because of this, the person is suffering from hemophilia. So remember this concept of hemophilia, the number of blood cells and how it is. Again, apart from hemophilia, now hemophilia is a genetic disorder. Is there any other disease that people suffer from because of the low platelet count?
Or low platelet dengue?
Dengue fever. So no, hemorrhage is a different thing. Hemorrhage is a different thing. What is hemorrhage? Breakdown of the blood vessels inside the body and the loss of blood inside the body. Oozing out of the blood inside the body. Inside the body. Whenever there is a breakdown. Say suppose a person is suffering from hypertension. A person suffering from hypertension and the blood vessels are torn inside the body and there is oozing out of blood. That is hemorrhage. That is hemorrhage. So oozing out of the blood outside the body, that is not hemorrhage at all. So here, name the two diseased conditions. Name the two diseased conditions. Now here, the disorder or disease also hemophilia that you have to mention. Name the two conditions associated with platelets. What are the two conditions you are writing as per your syllabus? There are some few, but as per your syllabus you are writing, if it is a disease condition, then it is dengue fever. And if it is a disorder, then it is hemophilia. Right, hemophilia and dengue fever. Can you complete this small progress check there? There are quite a number of them now. Page 98. What are those two? Try this at home. See whether you are able to solve all these questions. You should be able to solve 2 or 3 progress checks are there.
This is serum or plasma? Okay, first one.
Plasma.
The yellow colored part of the blood, that is plasma. From where fibrinogen is removed, then serum. That is serum. Okay. Go through this part, the next class. Try this question, see whether you are able
Granular, not granular. WBC. WBC. Increase in WBC. Increase in cancer. I can't buy that. Cancer. See, it is not a normal increase. It is from abnormal increase in infections could be. So it is not necessary to go for yoga. So you can't follow that condition. That is my person. So yeah. हर एग्जाम में पहले रिजल्ट में पागल हो बैठ रहा हूं भाई ऐसे कोई नेक्स्ट नहीं मारना मैंने ऐसे ही निकाल दी है चलो ना चलो एक मिनट किसी फोटो दे दो बदतमीज लोगों को जल्दी आएंगे क्या लुंगी बोले तो इसके पागल अच्छे ऐसा कोई फ्लेक्स नहीं है बट देखते हैं मैं देखा तेरा स्लाइड हां शोरी के मैं बोल रहा हूं एक मिनट मेरा बोल देखा मैंने आपको वीडियो देखा अरे मैं देख लिया ओके वो अच्छा था यार